this kind introduction and the opportunity to share our experience with other colleagues using the GeneReader NGS system in daily routine and during our validation study. In my short talk, I want to give an overview about our Institute of Pathology and our formerly mentioned verification study. And at the end, I want to summarize our impressions and give a short conclusion. The Institute of Pathology is in the west of Austria, next to the Lake of Constance. It belongs to the federal state of Vorarlberg with 360,000 inhabitants. Here you can see me on my daily way to work. This is our new institute. It was built in 2008. On the upper floor you can see the laboratories of the molecular diagnostic. Our institutes includes the departments of histology, cytology, autopsy, microbiology, and also the molecular diagnostics. In our daily routine, we have to deal with a lot of different samples and sample types as liquid cytology, liquid biopsy, FFPE material, and so on. And we have to do a lot of molecular testing like pyrosequencing, real-time PCR, as well as immunohistochemistry and also FISH diagnostic. In modern medicine, the tumors are diagnosed in a very early stage so that we have to deal with low amount of tumor material with a growing number of molecular applications. So, to face this new challenge, we introduced the GeneReader Next Generation Sequencing System. Since May, we use the NGS system for detection of somatic mutations. At the moment, we are established the BRCA mutation as well as the fusion gene detection. I think everybody knows, but I want to mention the hardware components of the GeneReader. We have the QIAcube and the QIAsymphony for automatic DNA RNA extraction, the qubit for the quantification of nucleic acids, the QIAgility, we use this for automatic PCR setup to save some time, and some real-time PCR machine like the RotaGene. We also have the QIAxcel. It's for the quality control after target enrichment and library preparation. This is one big advantage of the system. And the GeneReader QIAcube, it's for the clonal amplification and the following steps, as well as the GeneReader sequencing system. For our in-house validation study or verification study, we use 2 Horizon controls, 50 FFPE samples analyzed by pyrosequence and real-time PCR during routine procedures in the years before, additional 20 liquid biopsies of patients with non-small cell lung cancer or colorectal cancer, and we use the Horizon controls for inter- and intra-assay variability. So, with the Horizon controls, this is controlled DNA, it's commercially available with a known mutational status. We analyze replicates of 20 of each control in 2 flow cells for the intra-assay variability. We repeat the same analysis in the following week for the inter-assay variability. And we get 100 concordance of all detected mutations in every run. And some variations in the detection of out-of-spec variants or run-reported variants were reanalyzed and were judged as background. Then we are using 15 FFPE samples. As I mentioned before, they are characterized by pyrosequence or real-time PCR for the mutational status. In 92% of the mutations detected by pyrosequencing or real-time PCR were also confirmed by the NGS runs. 8% of the samples had to be excluded due to low nucleic acid quality or poor sequencing quality. Additional mutations we were detected during this next generation sequencing analysis were later confirmed, for example, by pyrosequencing or other sequencing techniques. Now I want to come to the conclusion. In daily routine, the GeneReader provides a highly automated NGS system. It reduces hands-on time because we had a lot of machines that are doing the work for us. The hardware components are long on the market, so there are no teething trouble. And it's very important. One system in one hand. It's easy to troubleshoot because we have one company to get to. Another big advantage are the quality control steps after the crucial steps like the target enrichment or library preparation using the QIAxcel. It's a capillary electrophoresis. As I mentioned in the session before, I think you learn about the software. That's because I just give the conclusion. The GeneReader Assistant software allows a fast access to all relevant manuals, principles, and procedures during NGS workflow. The GeneReader Link software allows software-based sample tracking. You know every time where your sample is and what step. And the QCI Analyze software, it's a third software, provides quality control as well as the sequencing data. The QCI Interpret software is the fourth software. It uses or shows the detected mutations in a clinical context. So you can see it's a pathogen mutation or it's a benign mutation. Now we are working with a customized generating report together with QIAGEN. It provides characterization of the detected mutations, gives an overview about the variant details, the reference, approved therapies, and clinical trials, and so on. Thank you.
QIAGEN.
Sample to insight.